In our previous lecture of genetics, we talked about the Mendels laws. Our today's topic of the lesson, principles of inheritance, are incomplete dominance, codominance, multiple allelism, polygenic inheritance, and pleiotropy. So, dear student, incomplete dominance is exception of Mendel's first law of dominance. It is the phenomena of neither of two alleles of a gene being dominant over each other, so that when both of them are present together, a new phenotype is formed with somewhat intermediate phenotype between the independent expression of two alleles. This phenomena was discovered by CODOMINANCE. Let us take an example. A plant named as Antirrhinium majus, also called as snapdragon or dog flower. There are two types of pure breeding plant, red flowered and white flower. When they were crossed in F1 generation, they do not have red or white flowers. Rather, they were having some different phenotype called pink flower, which was due to interaction of two alleles of red and white flowers. This is no blending of two alleles, and effect was due to incomplete dominance of alleles of red color over white color. When F1 pink plant were selved, in F2 generation, three type of plant were produced: red flower, pink flowered, and white flowered in the ratio of 1:2:1, and both genotypic as well as phenotypic ratio was same, that is 1:2:1. Two plants are the example Mirabilis jalapa and Andalusian fowl along with Antirrhinium majus. Let us focus on first. A red flower plant can be represented by RR and white as RR. These were parents, gamete produced by R and R. They fused, and in F1 generation, plant produced were pink, somewhat intermediate between red and white. Though it was expected that plant must be either red or white, but they were pink due to incomplete dominance. When these pink F1 were selved, both pink plant were selved in F2 generation. Due to checkerboard, let us study it. RR were the gametes. The phenotypic as well as genotypic ratio was found same, that is 1:2:1, and three type of phenotypic plant were produced, red and pink and white, in 1:2:1, which was different than that of expected 3:1 of monohybrid cross of F2. Here red, pink and white, three type of flower were produced. This clearly shows that the red color producing gene was not dominant over white, rather it was incompletely dominant over white. Just when keep in mind, phenotypic and genotypic ratio 1:2:1 is same only and only in one case, and that case is of incomplete dominance. Another example is of Andalusian fowl, where black and white were crossed together, black coat color and white coat color were crossed, and in F1 generation the organism produced were of blue coat color. When these F1 were selved, in F2 generation, black, blue and white, three type of organism were produced in 1:2:1, and the genotypic ratio was BB, BB and BB, 1:2:1. Checkerboard is drawn here. BB was crossed with BB, F1 selved, the gamete produced were B and B, B and B, and F2 generation, when these two come together, it was formed like this. If these two come together, it will be blue. If these two come together, it will be white, and if these two come together, it will be blue. The next topic is codominance. When two alleles are equally dominant and none of them is dominant or recessive, the phenomena is called as codominance. So the phenomena of two contrasting alleles of same gene lacking dominant recessive relationship and expressing their effect simultaneously in a heterozygous condition is called as codominance. Codominance can also be defined as when the two alleles present on homologous chromosomes neither show dominant recessive relationship nor show intermediate condition, but both of them express themselves simultaneously. The condition is called codominance. It is most common in blood group in human being. See, the blood group A, when crossed with blood group B, the alleles are IA and IB. This is B blood group and this one is A blood group. When a person with blood group A marries with a person with blood group B, the gamete produced are IA and IB, and their F1 generation is neither having A blood group nor having B blood group, rather some intermediate blood group AB is produced. This shows that both allele A and B have expressed themselves in F1 generation. Same example we can take for sickle cell anemia. A patient have normal hemoglobin written as HbA producing gene as well as sickle shaped hemoglobin producing gene HbS. Both they show their expression. If a person has HbA HbS genotype, it means it's 50% hemoglobin is of normal shape and 50% hemoglobin produces RBC, that of sickle shaped. The next term is multiple allelism. As we are aware that for each character there are two alleles, but in case of multiple alleles in a population there may be more than one allele of same character. So multiple alleles are alleles of same gene which occur in the population of a species. All of them are found on same gene locus on the same chromosome or its homologue. However, a chromosome has only one allele and a diploid individual have only two alleles. We will discuss characteristic of multiple alleles. They are more than two forms of the same gene. All are mutant of same wild type. Out of various alleles, one is dominant over the other and other is recessive to all alleles. A gamete carries only one allele at a time. An individual has only two alleles of a gene. A very good example of multiple allelism is ABO blood group in human being, which is controlled by a gene capital I. Plasma membrane of RBCs has sugar polymer producing form from its surface and kind of sugar is controlled by a gene. Gene I has three alleles, IA, IB and small I. IA and IB are dominant over small I. Allele I and IB produce slightly different form of sugars while small I does not produce any sugar. As human are 2N, that is diploid, each human will have any two of the three I genes alleles. IA and IB are dominant over small I, that is if IA and small I are present in an individual, the blood group will be A because IA will express. Since there are three different alleles and six different combinations of these alleles are possible, which are called as six genotypes. So there are four phenotypes and six genotypes in human population for blood group. In other words, we can say there are four blood group only, A, B, AB and O, but the A blood group can be produced in two ways. IA, it is called as homozygous dominant, while IA, small I is heterozygous. Similarly, B blood group can be produced in two ways, IB, which is called homozygous, and IB, small I, called as heterozygous. And the next genotype is IAIB, which is heterozygous, and the last genotype is small I. Thus, there are six genotypes, 1, 2, 3, 4, 5 and 6. And four phenotypes, that is A blood group, B blood group, AB blood group and O blood group. Another topic here we are studying now is polygenic inheritance or polygenic trait. So far we have talked about for each character there is a one gene. However, it may possible that a trait may be controlled by more than one gene. Such an inheritance is called as polygenic inheritance or polygenic traits, and those traits which are controlled by three or more genes are called as polygenic traits, and the inheritance is called polygenic inheritance. Each gene of a polygenic system contribute to the phenotype but to small degree, and presence of more than one dominant gene makes phenotype more prominent. In other words, all dominant genes adapt their effect to produce full phenotype. A very good example of polygenic inheritance is skin color in human being, where phenotype reflect the contribution of each allele and is additive. Let us take there are three genes A, B and C, which are capital letters, means they are dominant, which control skin color in human with dominant forms capital A, capital B and capital C, which are responsible for dark skin color, and recessive allele, that is small a, small b and small c, are responsible for light skin color. The genotype with all dominant allele, that is AABBCC all capital letter, will have darkest skin color, while recessive alleles AA, BB, CC will have lightest skin color, and remaining intermediate forms, this AA, BB and CC will have an intermediate skin color. And a person with 3 dominant 3 and 3 recessive allele will have intermediate skin color, which will be represented by like this IA, that is AA, BB and CC. Hence many genotypes and phenotype are possible. That's why two organisms, two human beings in same location do not have same skin color due to presence of these allele as well as due to their environmental interaction. The another term is pleiotropy. Previously we talked about polygenic inheritance, which means many gene control one trait, one character. Now in pleiotropy we will discuss in reverse, means more than one traits will be controlled by one gene. So the phenomena of same gene controlling the expression of a number of traits, hence single gene can exhibit multiple phenotypic expression, is called as pleiotropy. A very good example of pleiotropy is a disease called as PKU, that is called as phenylketonuria. So there is a disease called as phenylketonuria in human, is caused by mutation in the gene that codes for enzyme phenylalanine hydroxylase. The name of enzyme is phenylalanine hydroxylase, which is responsible for digestion of an amino acid called as phenylalanine. This manifests itself through phenotypic expression like mental retardedness, reduction in hair and skin pigmentation. Here one gene is controlling multiple metabolic function of enzyme and hence is called as pleiotropy. Another example is starch synthesis in pea seeds is controlled by one gene with 2 alleles, capital B and small b. Starch is effectively synthesized by capital B with large starch grain, while small b have less efficiency of synthesis with small starch grains, and capital B small b, that is intermediate, will have intermediate size of starch in grains. Now the next topic is limitations of Mendels Law of independent assortment that we have studied earlier, which separate the traits in 9:3:3:1 ratio. It is true only for those alleles which are present on different chromosomes. In case genes are present on same chromosome, they would be inherited together and phenomena will be called as linkage. Then we come to what was the reason for great success of Mendel in his experiments on pea plants. First point, he selected only pure breeding varieties. He selected 7 characters for experiment after confirming that they did not show any linkage. He worked on 1 to 3 characters at a time. Next point, varieties selected by him had well-defined contrasting traits for a character. Pea plant is ideal for breeding experiment like emasculation, bagging and tagging because the flower size is large. He emasculated flowers in early bud condition. He applied statistical analysis for his results of experiment and he had large sample size to avoid chances of error. So dear students, we studied today incomplete dominance, codominance, multiple allelism, polygenic inheritance, pleiotropy and reasons for success of Mendel. So I hope you have studied and understood all the topics. In coming video we will discuss about sex determination mechanism and chromosomal theory of inheritance.